Okay, now let's come to the four major receptor classes or families that are available inside the body. We said that the receptors can be a broad term of macromolecules to which they are targets for drug binding. But there are four major classes of receptors that we should know about as future medical doctors. So the first one of these major classes is the ligand-gated ion channels, which is the fastest, actually. The fastest type, carries the fastest type of response, such as cholinergic nicotinic receptors that are available on the neuromuscular junction, putting in mind that the neuromuscular junction needs to act very fast. Therefore, this receptor on the cell membrane, by binding of the neurotransmitter here, the triangular form that we see, is the example of the acetylcholine at the neuromuscular junction released from the motor neuron, and then upon binding, it opens the ion channel. That's why the receptor is called ligand-gated ion channels. As we said, a few milliseconds will be enough to produce the response of influx of sodium and efflux of potassium, which is a ionic exchange behavior of that receptor when it's activated. The second one is a G protein coupled receptor, a bit slower than the first one. Uh, as the name implies, it's coupled to G protein upon activation of that receptor by binding with the drug. It leads to detachment of certain subunits of the G protein intracellularly on the intracellular part of the cell of the receptor, and then this detachment will lead to activation of second messengers, as we have mentioned before in the previous slides. Now, for this type of receptor, we know that the response lasts several seconds to minutes, in contrast to the fastest one, which is the ligand-gated ion channel. The third type of the major four receptor types is the enzyme-linked receptor, and we gave the example of insulin. And here, the binding of the insulin as a hormone to the cell membrane receptor will lead on the intracellular part to the activation of tyrosine kinase, which is the enzyme that is linked to this receptor from the other side of the cell membrane. And this will lead to the biological response of insulin, whatever it is, by reducing blood sugar or stimulation of the synthesis of glycogen in the liver, whatever the biological response of insulin, depending on the organ. The last, or the fourth type of the major class of receptor families is the intracellular and actually intranuclear receptors, such as the steroid receptors and thyroid hormone receptors, thyroxine receptors. Here, because of the position of those receptors intranuclearly inside the nucleus, so the activation and response will take hours to days because the drug need to penetrate through the cellular membranes. GABA-A here is the example of a ligand-gated ion channel. GABA is gamma-aminobutyric acid, which is the major inhibitory neurotransmitter in the brain and the CNS. Yeah, as you can see, it is a cell membrane receptor, as we mentioned, and it is a ligand-gated ion channel. The ligand is GABA itself, gamma-aminobutyric acid, the neurotransmitter, which upon binding to the active binding site, it leads, the active binding site is located between the beta and the alpha subunit. Two molecules bind at the same time, lead to opening of the chloride channel. So it's a ligand gated ion channel. The opening, the activation of GABA to the GABA receptors chloride channel will enhance the GABA mediated chloride influx. So this influx will lead to increased intracellular chloride negative ion, and the negative ion intracellularly will lead to hyperpolarization of the cell membrane. This means that the cell membrane will be stabilized, and that's why GABA and its receptor are considered as neurosuppressants. They are they suppress the activity of the neuronal impulse initiation and propagation through the central nervous system. That's why they are considered as CNS depressants. Now we can see that there are other receptor sites for benzodiazepines, flumazenil, and these are considered as allosteric binding sites. Some of them are allosteric inhibitors, others are allosteric activators, such as benzodiazepine. So this is the example. There are many other examples. We mentioned the nicotinic receptors also at the neuromuscular junction, and here is another example, which is GABA receptor as ligand-gated ion channels. The second example for the G protein coupled receptor is the beta-2 receptors. If we take the airway smooth muscles as the tissue of example, in the airway smooth muscles, they are full of beta-2 receptors. Beta-2 adrenergic, they are sympathetic receptors, to which adrenaline can bind as an endogenous neurotransmitter, or if we're talking about drugs, then salbutamol is one of the major examples that are used as bronchodilator. Salbutamol is used as a bronchodilator because it can stimulate the beta-2 cell membrane receptor, and by binding of cell membrane receptor, this is a G protein coupled receptor. This is a G protein coupled receptor, as we have mentioned before in the description of the first slide about the 4 major types. And then by stimulation of G protein, the second messenger will be the cyclic AMP. So what will happen is the stimulation of G protein on the intercellular part will lead to activation of cyclic AMP from ATP, conversion of ATP into cyclic AMP. The increase in cyclic AMP will lead to activation of the protein kinase A. This will lead to smooth muscle, airway smooth muscle, cellular relaxation. So this cellular relaxation will lead to bronchodilation. Now the other example of that increase that also can cause the increase in cyclic AMP and eventual bronchodilation is the phosphodiesterase enzyme inhibitor, which is the phosphodiesterase is the enzyme that metabolizes cyclic AMP. By inhibition of this enzyme, metabolizing enzyme, the ultimate effect will be increased cyclic AMP2. So the example is theophylline and aminophylline as examples of phosphodiesterase isoenzyme 4 inhibitors. The ultimate outcome will be increased cyclic AMP and bronchodilation. Both of these drugs can be used effectively for the treatment of bronchial asthma, and especially the bronchoconstriction attacks during the acute attack, to relieve the bronchoconstriction by increasing cyclic AMP and ultimately leading to the bronchodilation. Now the third type, the example of the third type, as we mentioned, is insulin receptor, which is enzyme-linked receptor. By binding of the insulin to the cell membrane receptor, this is a cell membrane receptor which is inactive, but upon binding it will lead to activation of tyrosine kinase enzyme, and the activation of tyrosine kinase enzyme will lead to activation of multiple signaling pathways, as we mentioned, that lead to intracellular movement of glucose from the blood into the different tissues such as skeletal muscles and other organs, so that it will reduce the blood sugar. But taking into account the behavior of the enzyme, the slow behavior of the enzyme, and requiring time, it lasts longer time that is than the previous two examples, the previous two classes, which are the ligand-gated ion channels and the G protein coupled. So this will last for minutes up to hours in order for the blood glucose and the action of the insulin to take place, due to the fact that it needs to activate a tyrosine kinase intracellularly, which will take longer time. The fourth and the last one is the steroid receptor as an example of an intracellular intranuclear receptor. So any drug that needs to act on this receptor intranuclear to make uh, translational changes for specific proteins to be synthesized or to be inhibited, whether to be synthesized, over synthesized, or to be inhibited. These drugs need to penetrate through the cell membrane and penetrate also through the nuclear membrane. This requires the drug to be sufficiently lipid soluble. Sufficiently lipid soluble. Water soluble drug cannot do that. Only lipid soluble drugs are able to diffuse through the cell membrane and the nuclear membrane in order to reach the receptors, through intracellular intranuclear receptors. And therefore, because of the position, the distant position, the far position inside the nucleus, the activation and response of these receptors will take the orders of hours and days, such as steroid hormone and thyroxine as an example of these hormones also.